Is this one?
Yeah, you need it in the USB.
Oh! Okay.
I think it's in here.
Oh, yeah, but I wanted the pointer. The pointer is the
Let
right
me
one.
see. Let me see. I don't know exactly how it connects.
But doesn't matter. But the pointer is the right one?
Yeah, the pointer is connected.
The thing that is there.
Let me see.
What has an on button?
Here's the power switch.
Okay.
Si no lo pones en marcha,
No.
works also.
Unexpectedly, yes. I thought the speaker was on. Okay, thanks.
Well, good afternoon, everybody. It's a pleasure for me to introduce you Jordi Soriano, who studied physics in the University of Barcelona, where also did a PhD degree there. And after that, he did two postdoctoral stays. The first one at the university, let me pronounce it well, because we have a German here, of Bayreuth, in Germany, not Bayreuth, but Bayreuth, in Germany. Where he studied the regeneration process of freshwater polyp iron. And then 2 years, he spent 2 more years in the Weizmann Institute in Israel, studying already connectivity in living or in neuronal systems, starting to work on that. And since 2008, he has a Ramón y Cajal contract at the University of Barcelona, where he's starting to set up a very nice and funny, I would say, laboratory for the generation of cultures and build connectivity in cultures of neurons. And I also heard that he is negotiating, I'm not sure if I can say that, but I heard anyway, negotiating an European Research Council starting grant, which means that he has a very high level as a scientist. So I hope you succeed with it, and thank you very much for coming.
Thank you. So yes, you can say that. Yeah, it's very nice that I have the opportunity to defend my ideas in Brussels in about 3 weeks. So let's hope that it works. And then we will organize a super seminar to celebrate it. Okay. So basically, yes, I want to talk today about the experiments that I started at the Department Institute about 4 years ago, more or less now, where I was working in different techniques to try to get information about the connectivity in neural cultures in an easy way. I will go on that later. And to start discussing about that, I always like to mention a little bit why neuroscience is so nice for most of us. So those are maybe 5 of the most interesting problems nowadays in neuroscience. So let's start with computing. Computing means that people want to understand how just the nice wiring between neurons in the brain is able to produce such nice things like thought, emotion, love, intent. So it's very, very complicated. Why the brain is so complex and how that complexity forms ourselves. The language is something very interesting. Only if you travel, for instance, to China, then you realize how difficult it is to communicate with people. But when you think about that, it's very fascinating that we are able to code and decode information just by making noises from our mouths, which is something very, very fascinating, that you have to synchronize very well different parts of the brain, plus many, many muscles in your mouth. Another thing that is interesting is memory and learning. We are able to remember things, or learn things in milliseconds, for instance, that hot things are very nasty, and we can remember that for our entire life. And it's incredible that we have such a sophisticated system to record information in such a way and forever. And then a thing that is very funny and indeed fascinating to read papers about that is why we have to sleep and dream. And people are still debating about that. And the current opinion is that we need somehow to reset the senses that we don't need to have the system ready for the next input of data. Okay? And of course, I think that is between physics and, let's say, matrix is the networks problem. This more or less what is common between us, and it's how the brain is built itself, the architecture of the brain, and how that evolved to have a human being like us, and that is indeed so complicated. So what is our problem, to be more specific? Well, you know that the brain is a very powerful and an incredible machine. We have typically 10 to 11 neurons and 10 to 14 connections, and we lose about between 10 and 100 million neurons per year, more or less. This is our rate of death. But don't worry, because we have a lot of them to keep going on. But I think that is interesting, and people are not very aware about that, is that the brain is first constructed using a powerful genetic machinery, but the idea is that basically the connections are just put in place roughly. This is something that's surprising, but there is not enough genetic information to locate precisely the neural circuits. What happens is that there is a strong refinement in the form first of spontaneous activity in the brain, and later from sensory experience. A typical example is what is called the blind cat. So basically, if a cat is born in a dark room and doesn't see light for a few days, the cat will be blind. And the reason is that if the visual cortex doesn't get information from the eyes, doesn't know how to see. It's surprising, but we need to learn. We have to get input from the outside to be able to refine the neural circuits to do what they have to do. Okay, we have to talk, we have to learn to talk, we have to learn to walk, to see, and to hear. The interesting thing is that, as you will see now, neural cultures don't have all this genetic layout, but they have some activity and some possibilities to verify it. One of the problems that neuroscientists face today is that we have really problems to try to understand how all this wiring is formed. I mean, you know that there are so many neurons and so many connections that to get the details of the precise wiring between two neurons is extremely difficult. Extremely difficult. And now one needs very complicated techniques that are normally very slow and require a lot of thinking. So our objective advisement was to think, okay, can we devise techniques to get information about the connectivity of a neural network that is not so painful, let's say? And the response is yes, that we work in that direction, and it's basically what I'm going to explain you now. In any case, if you're curious and you want to know a bit about the current techniques to get the precise mapping of the brain, you can take a look to this nice paper of Nature, this paper quite recent, that explains all the machinery that we have in the labs today to get exactly that precision. As far as I know, there is full mapping only for the C. elegans. This is a very famous one, and also for some parts of the cortex of the mouse, the cat, and the macaque monkey. So neural cultures is what we use in our lab, and it's something very simple. You just take the brain of an embryonic rat, typically 17, 19 days of gestation. Normally a rat gets birth about day 21, so they are very mature brains, so they are actually already formed, but the neurons are basically disconnected. So it's very easy to get the brain, dissociate the neurons, and then play them in a glass cover. So this is basically what happens. And to get an impression, the typical size of a neural culture is more or less the area of the brain, basically. So in this area, you can easily allocate 1 million neurons, okay? So what happens is that neurons are plated at day 0, and they immediately start to form connections between them, and they form a mature culture in about 2 weeks. Typically, neurons are kept alive in a culture well like this. So this is, let's say, the glass where they are sitting, and then if you put nutrients, vitamins, and everything they need, they can survive for typically 2 weeks to about 6 months. 6 months is a record today. And even people is working on methods to try to have living neural cultures for years. Actually. So this is the typical culture that one can get in the lab. So those black dots are neurons. It is more or less a homogeneous distribution, and this is a distribution with clusters. These are big blocks of neurons. There are easily 1,000 neurons here, and these kind of wires that you see here, these are highways of axons. These are hundreds or even thousands of axons packed together. Okay? So the experiment that we have actually thought at Weizmann is the following. Let's have a culture like this one. Now let's mark all of them. Okay? And now let's put an electric excitation.
Are these individual ones or clusters?
Oh, these ones? Oh, these are individual neurons.
Okay.
So let's mark individual neurons. We can typically choose about 1,000. Note that if you try to record the same, the activity of a single neuron using other methods, nowadays the common ones are what is called patch clamp, which is to put a pipette in a neuron and measure directly the voltage. Another one is called multi-electrode arrays, and is to put an electrode, a battery of electrodes in the culture, and then try to measure the activity in each electrode. But the problem is that for patch clamp, the record today is 7 patch clamp at the same time, and for multi-electrode arrays, the record is 64. So if you want to get more neurons, you have to go to this technique, which is called fluorescence imaging. So the idea is that you select the neurons, and then you use a fluorescent dye that indicates when a neuron is firing by a sudden increase in the light that you are observing. Okay? Okay. So basically what we do is we select all the neurons that we can see in the field of view, and then we zap them. We apply an electric field to the entire culture, and then we observe how neurons react to the external excitation, and after that we reduce gradually the connectivity of the network to see how the network degrades as we excite it. I will go on details right now, but the central idea is this one. We have the neural culture, we monitor individual neurons, we apply an electric field, as shown here, and we record the fluorescence of the individual neurons as we excite them.
Sorry, Jordi, what's the time resolution of that?
Yeah, the typical time resolution is about 200 milliseconds. We can go to better resolution, and we're improving that now. But the point is that in this kind of experiments, we don't care about dynamics. It's not interesting for us. We just want to get information about the connectivity of the network, and because we ignore the dynamics for the moment, we just want to know at which moment neurons start to fire when you excite them, but not what happens later.
Yeah, that's a little piece of your whole culture. So the places where you actually apply this field are far away.
Yeah. We apply the electrodes across the entire culture. Yes, but we are monitoring only about 10%. But because these neurons are connected to all the others, basically you can get good statistical information about what's going on the entire culture just by looking at a small detail of it.
Another question. In one previous slide, you showed these big clusters. So that's a very late stage, or?
This? Oh no, because this is because you can prepare the substrate. You can play a lot with the substrate. You can normally you play the neurons on a glass covered slip. If the glass is extremely clean, then they form this homogeneous distribution. If the glass is dirty, or you intentionally put regions that are different, for example, with some biochemical treatment, then you can get this kind of cluster neurons. So basically, this is how the setup looks like. So basically, this is the lamp that is giving the fluorescent signal, and more or less, you see this is the entire culture, and the blue area is basically what you are actually measuring. Okay, and you can see here the two electrodes. This is to give an impression of the size of the sample. Okay. And this is an example of the dynamics of the network. So you see, they fire. Well, maybe I'll do it again. So this, oh, thanks. So this is actual recording, and now you will see that suddenly there is an increase. Okay? So this is the activity of the neurons. All neurons fire at unison in a synchronized manner, as you see in the plot on the top, that is the recording of each individual neuron. Okay? I will go on that later. But what I want to describe is this technique, and for that I insist that we excite the neurons all together. At the same time, we monitor the neurons. But the fascinating thing, the important thing, is that we reduce the synaptic strength between the neurons. You know that for a neuron to fire, the neuron needs to receive inputs from other neurons. So the electric signal coming from an axon arrives to the synaptic terminal in this way, and then the electric signal is converted into chemical signal, which in form of vesicles transports the neurotransmitters from one cell, from one synaptic end to the other one. The transmitters are collected in the receptors, and then those receptors modify the properties of synapses to release an electric signal, and this is the way the signal is transmitted. So the important thing is that one can block the receptors at the postsynaptic gap to reduce the efficiency in the transport of the vesicles, and therefore reducing the communication strength between the neurons. Okay? This is a standard method. So one can ultimately completely block the communication between neurons. Okay? So there are many receptors, there are many antagonists, and to put a little of order on that, one can basically reduce all the problem to only this family of receptors and antagonists. So basically, receptors is the largest family. AMPA receptors is the largest family for excitatory neurons. I don't know if you're familiar with that, but typically you can find two kinds of neurons, excitatory and inhibitory. The excitatory neurons, what they do is to increase the membrane potential in a neuron, while the inhibitory ones reduce the potential. And only when the top potential arrives to a threshold, the neuron fires. Okay? Are you familiar with this, more or less? Well, but this is the general concept. So basically what we do in our experiments is we'll consider only AMPA receptors. And we will apply sync-to-X to gradually reduce the connectivity of the neural network. Okay? To avoid further complications, what we do is we block completely all the other receptors that are non-AMPA with APD. Okay? So basically we have only excitatory neurons with AMPA receptors. And what happens with inhibition? Well, we can do two things. We can either leave it active or block it completely. And as we will see later, this is very interesting, because by playing with one or the other, we can get information about the role of inhibition in the network. Okay? Are you following more or less the idea? So now we go to the real experiment. These are actual measurements. So to understand it, let's start with this easier situation in which neurons are completely saturated with sync-to-X. This means that neurons cannot communicate with each other, cannot pass an electric signal. So basically they respond only to the external excitation. So we excite the neurons, and then we see that only a few neurons respond. Okay? And basically what we observe in the experiments is that if we scan in voltages the sample, we observe that at low voltages only few neurons fire. At more voltages, more neurons fire. So basically we got this nice error function curve, which indicates that basically the distribution of excitability of the neurons is Gaussian. Okay? And this is very, yeah.
This is already from the coupled neurons, right? Do you know what the threshold?
For completely disconnected neurons.
These are disconnected?
Yeah, these are completely disconnected neurons.
Okay.
Yeah? So that you have to scan basically one by one to excite the neuron. Okay? But the important thing is that, because this is the behavior in excitability, you have to excite only the neurons with the lowest threshold to light up the entire network when it is connected. Okay? So this is completely disconnected network, and this is completely connected. Sync-to-X equals 0. So here nothing happens, and then suddenly all light up. Okay? One extreme, the other extreme. But the interesting thing is what happens in the middle. And in the middle we observe something very interesting. And it's that as we increase a little bit the concentration of sync-to-X, which is this value of 100 here, and we see that there are small regions where only few neurons fire, that they form small clusters. Okay? either at low voltages or at high voltages, but there is a characteristic large jump that indicates that there is a population of neurons that is strongly connected. Okay? We call it this giant component, and indicates that there is a special cluster on the network that connects most of the neurons. Okay? And this already suggests that there is some kind of percolation transition in this problem. Okay? We increase more and more the concentration of the drug, the giant component gradually reduces in size until finally, for around some critical value, in this case 1000 nanomolar of sync-QX, we don't see any characteristic jump on the system. Okay? And that indicates that possibly the percolation transition occurs around here. Okay? Something that is very fascinating in these problems, and is that we can actually wash the sample, we can actually take out all the drugs and repeat the experiment, and we recover exactly the initial network. Okay? The bad thing is that neurons don't like the fluorescence imaging, and they die after 10 hours. But in 10 hours, you have enough time to run a few experiments. Okay? But you understand the concept of the problem. We use electrical excitation to excite individually the neurons and see how many neurons responds when one responds, and if there is any giant component in the system. So the first analysis that we did was to see, okay, we have this kind of, we have this nice behavior. So now let's try to do some data analysis. And the first data analysis that we did was to put in the same color the neurons that were firing together. So for low values of the drug, you see all the network is connected, and as the giant component decreases in size, the area that covers the giant component is continuum, as you see here, but finally at the percolation transition it breaks off in small disconnected clusters. Okay? What is interesting for us is that the giant component covers a continuum of the sample, and this indicates that neurons maybe, according to this result at the end, are connected to basically to the neighbors, but that there is still some long-range connectivity. Okay? And this is already something interesting. We are getting some information about the structure of the network with somehow an easy way. But we wanted to go a bit further, and for that we introduced a model that is based on percolation, but in particular is what is known as quorum percolation. Quorum percolation means that you need several inputs to connect with an entity. Okay? In this case, we define a control parameter that we call M, and M is the number of inputs required for a neuron to fire. And I will explain this in detail. Imagine that the system is so simple that we basically have the membrane potentials, which is typically about 30 millivolts, and then the contribution for each single synapse, which is about 2 millivolts. This means that if we don't touch anything on the culture, typically a culture will need about 15 inputs from the neighbors to light up. Okay? What happens? Well, this is the synaptic strength between the neurons, and we know that sync-QX weakens that strength. And actually, one can do a bit of biochemistry and realizes that the relation between the strength and the concentration of sync-QX is given with this equation. So basically, this parameter M increases with the amount of sync-QX, starting from a value M0 that, as I said before, is around 15. Okay? So basically, we have our culture, we don't touch anything, and neurons are very happy, and they just need about 15 inputs to fire and pass the signal. Yes?
The theory is nice, but we are assuming that all the inputs arrive at the same time, although you are not resolving the dynamics. Exactly. You have to assume that they are arriving at the same time.
Exactly. We are assuming many things. Now I will put a list of assumptions that we have. It's true, we completely ignore dynamics, and basically we observed that the results were so good, the model was so good compared with the experiments, that we thought that we really had something very interesting. But it's true, we ignore too many things. And it's incredible that such a simple model works so well. I will show now the results. But basically, to understand this control parameter, I put just this simple cartoon. In this case, this doesn't want to represent any real number, okay? It's just a cartoon. where I have n0 equal 2 and an average connectivity of about 4. Okay? So at the beginning, if I don't touch anything, no drugs, m equals m0 and is equal to 2. Okay? So any neuron that has at least 2 neighbors will fire and pass the signal. Okay? So in this case, all the network is connected. But as I increase the amount of drag and m goes up to 3, then neurons that don't have 3 inputs will not fire and therefore will get disconnected from the network. So only this pink area here is now the giant component, and so on. And we realize that typically for a value of m that is on the order of the average connectivity, the giant component breaks off and you have only isolated clusters. Okay? And for very large values of m, infinite sin to x, the network is completely disconnected. Okay? This is the concept. And this is what you want to come here. So basically what we wanted to do is to see if a very simple percolation model is enough to just describe the connectivity of the network. And we just wanted the minimum ingredients and with the simplest mathematical description. And yes, and we ignore the dynamics, so neurons are integrate and fire that we assume that get all the inputs simultaneously. And we can deal only with the input distribution. We can get information only about the inputs that get the neurons, but we cannot say anything about the length of the axons, for example. An axon can be very long, up to 1 millimeter, typically, but we cannot provide information about that. We have to combine this technique with others, for instance, immunostaining, GFP, to get the actual shape of the axon on the culture. And then we also have something important, and is that connectivity is described as a graph, in the sense that neurons are somehow in a topological space, and the distance between the neurons doesn't matter. Okay? And of course, as I said before, we ignore the axonal tree. I mean, it's huge. Yeah, I mean, it's a huge amount of assumptions, but the funny thing is that it works very well, and it's here. So if I first consider the original plot, where we have the different curves as a function of the drag, and now I eliminate the drag and I use this other parameter, quantum parameter that I call m, then we have that the giant component decreases steadily until a critical value that gives me information about the average connectivity of the network. Okay? So basically what we did was to compare the experiment with numerical simulations using such basic model, and we got a very nice collective agreement within both of them, which is quite impressive for such a simple model.
Did you assume that all the neurons have the same frequency, pulsating frequency?
Pulsating frequency? What do you mean?
Well, the period of the neurons, when you put the parameters into the integrated and fire model, that has depending on the parameters, that has a typical period of pulsation. Did you assume the same for all of them?
We don't even use that. We don't even use that. We just consider a pure network model.
Oh.
Neurons do not behave as an integrated and fire system.
I see.
They are pure nodes and links. And this is something that we think is very interesting, and we can use it to get interesting information about the network. So for instance, what is fascinating for us is that we tried different distributions for the connectivity of the network. I mean, even if you have a very simple network graph model, you have to make some assumptions about the connectivity of the network. So we tried a few of them. We tried Gaussian, we tried exponential, we tried power law, but only the Gaussian distribution gave a similar curve family as the experiments. And we went further, and what we did was to compare the decay of the giant component that we get from the simulations with the model. Okay, and in this case we use 3 different densities. So the dots are the experiments, and the red line are the simulations. At the moment, ignore the blue line at the moment. And we observed that we could fit very nicely the experiments with the simulations only when we used a Gaussian distribution. Okay? And basically the important thing for us now is that the fit gives us information about the average connectivity of the network. This is something very, very fascinating, because you have to understand that to get the value of the connectivity of a single neuron for a biologist is very high. So you have here an experiment that somehow in an easy way is telling you that for high densities you can get about 100 connections per neuron, which is a value that is very reasonable. And our friends in biology said that it's okay. I mean, they tried, they did some alternative measurements using electrophysiology, and they said, well, you're right, with this very simple model, you get numbers that are quite okay. And that made us very happy. And we went a bit further. And here is where we introduce the concept of inhibition. So as I said before, you can completely block inhibition with bicuculin, and in that case you get this blue curve, and this is the critical point where you have only excitatory neurons. And then when you have excitation and inhibition, what happens? Well, on average, neurons see less connections. Why? Because some of them are inhibitory. Okay, and because the total amount is what matters, when you have inhibition, then the network is somehow weaker. So you get basically the same behavior, but shifted to lower values of n. And of course, it's really nice because then you can compare the two critical points and get information about the ratio between excitation and inhibition in the network. Actually, this ratio is extremely hard to get. It's very difficult using electrophysiology. But here we have a nice method that uses simple physics if you want, but that provides interesting information. And that's not all. I mean, because one can even go further and say, okay, this is very nice, but actually the network, when the network is developing, it's also a percolation problem, because you have independent neurons at the beginning, at day 0, that start to connect slowly to the neighbors to form small clusters until finally they form a giant component. So we like that, so we did the same. We studied the problem again, measuring every 4 hours for about 3 days. And if you take into account that I was the only experimentalist doing that, you can understand how funny it is when you run an experiment for about 48 hours in a row and you went to sleep and you don't have time. It's very funny. But the experiment worked very well. And what happened is that the first 3 days after plating, neurons are silent. They need time to get in the new environment and get excitable to the external excitation. So they need about this time to be ready to be excited with the electric pulse. But after 3 days and 4 hours, we can start getting the distribution of independent neurons. And then as time goes on, typically at 12 hours, there is already the emergence of a small jump that gradually grows until a giant component appears just in 24 hours. Okay? So it's incredible, but apparently the network is just completely connected in 24 hours. And we know that this is something very beautiful because in the brain, it seems that it's also correct. For instance, at least in the mouse brain, as I said before, the rat normally gets birth around day 21. I was going to say appear, but no. Appears, let's say, at day 21. And people told us, biologists, our friends said that, well, if they monitor the evolution of a rat embryo, they observe that between day 19, 20 to 21, there is a huge change in the behavior of the formation of connectivity, and basically the rat is ready to be outside to the world and start getting information just before birth. Something very interesting, that indeed something very crucial happens the last 24 hours of the evolution of an embryonic decade. Okay?
Well, that's dangerous, because if they are born 2 days before, they cannot do anything.
Probably the rat would die, yes. Yes. It's dangerous, yes, it's true. But actually, the nature has mechanisms for that. And even if an embryo is born few hours or even 1 day before the normal time, the development still continues. Okay. Actually, there was a famous case for a baby, a human baby, for instance, that had one finger was missing, and during the first day after birth, the finger was deformed. Because the development doesn't stop until a few hours or days after birth.
Sorry? These cultures, when you say after 24 hours the whole giant component shows up, are you constantly pulsing during the 24 hours? Or does this happen even if there's no excitation of the whole thing?
No. I mean, we measure, well, the measurement takes only 10 minutes. Okay.
On the same culture you're doing the measurements. I mean, would the culture cluster even if you don't do any pulse within the 24 hours? Will it still cluster?
No, I mean, we can, because the culture can die in about 10 hours. What we do is that we can measure with the same culture about 3 curves. Okay? Yeah?
Yeah, my question is, would the curve still appear if you don't measure the third in the 3 hours, 12 hours, 16 hours?
Yeah.
Would it?
Yeah, I mean, you leave the culture and you don't disturb it, and then you measure after 24 hours, you mean that?
Yeah.
Yes.
Because you said you need like you said in the beginning, that the red stays blind if you don't give it optic.
Yeah, it's true. But in the case of neural cultures, you apply a trick. And the trick is that you keep them in a soup of nutrients, but also contains glutamate, and glutamate is a substance that excites the neurons constantly. Okay? So basically what you're doing is giving them information that there is activity around them to, you're cheating them somehow, no? But it's the only way that you can keep cultures alive. I mean, neurons have to sense that there is some activity in the network to be alive, otherwise they will start apoptosis.
It's activity, but they don't fire. So it's substantial activity.
No, it's sacrificial, they fire.
They fire? Then how do you measure that? So you are measuring the response to your
yes.
Or is the spontaneous activity what are you measuring?
No. I measure excited activity. I force them to fire. I excite them.
How do you distinguish you say that also spontaneous they are firing? So
yes. You can distinguish them because typically neurons fire but in bursts. So it's all the culture that lights up or is silent, and the typical interval is between 30 seconds and 1 minute. And in this case, the neuron, in this case, the network is not connected. So basically you have isolated clusters here. And if they fire very rarely, okay? And in this case, you have the entire network firing at unison. But in this case, when they fire, it's every 30 minutes, every 30 seconds or 1 minute. So you have a lot of time in the middle to excite them. It works very well. Yeah. I mean, it's something that is particular of neural cultures that we still don't understand very well, and it's why they are basically silent, most of them, and when they fire together, they fire in these collective bursts. Okay?
How is exactly the protocol of the experiment? So when you say you put a voltage, do you put that instantaneously? I mean, instantaneously, or do you ramp that?
No, you put the 2 wires. And the culture in the center. So you apply active field across the entire culture.
Yeah, but that you do instantaneously. So you go
from... Okay. No, okay. I understand. No, what we do is we give a pulse of about 2 volts for 10 milliseconds, and then the reverse one of minus 2 volts. Okay? So if the neuron fires, it fires. If the neuron don't fire, then we return to the original situation. Okay? It's a very short. It's a very short biphasic pulse like this. Okay, where it is 10 milliseconds maximum. Okay.
And the variation that you have is in the height of that.
And the amplitude of the amplitude is that one. So this is involved. This is in time. Yeah, that is the amplitude of the signal that we give to the sample. Actually, one thing that is important is that these experiments are extremely reproducible. This is something very surprising. I mean, we tried cultures from very different embryonic stages, okay, and all of them we got very, very similar behavior. Even if the neurons from time to time they fire, for us it's not important, okay? We can really follow this behavior and it's extremely reproducible. So now we go to more funny things. So as Claudia is complaining, we ignore completely dynamics. So now we are starting to work on that in Barcelona. So up to now that was my research advisement. And now in Barcelona I was very worried because we are not doing anything about dynamics. So we are trying now to do new things. And one thing that is particularly worrying us is why neurons fire in such on-off behavior. Okay, they are completely silent and then they fire all together, very synchronized, and then they silent again. So we're thinking, okay, can we do something to try to see if we can perturb a little bit the connectivity of the network and see if it is sufficient to change these dynamics? This was our essential working question. And what we do now in Barcelona is something funny, and is that we want to put the neurons in valleys of topographical modes made of PDMS. PDMS is a soft polymer that is like plastic, but it's very versatile, so you can make a lot of structures with that, even at micron scale. It's very impressive, the resolution that you have on that. So we are planning to prepare these kind of lines, for instance, to study propagation of a front along the line, but also to make 2D circuits and see the correlation between the topography of the circuit and the activity of the network. Okay? And then in the future, we will make more crazy things, and for instance, make three-dimensional patterns by connecting vertically axons between two plates. Okay? And so far, we're working on that. And on one hand, one-dimensional cultures was already studied by my colleagues' advisement for about 10 years now. And it's something that is very nice, very, very nice. And you can do a lot of crazy things. And I really recommend you to read this paper, this Nature Physics paper, because what they did was to build, using one-dimensional patterns, they built logic gates of all kinds. So they built a diode, this one. So basically, the axons have a very high probability to work, to grow in this direction and then cross to the next one, the next triangle, and so on. But one axon that goes this way ignores basically this small gap. Okay? So you can make directional connections. And then you can combine the lines with the diodes and make clocks. Because if you excite the neuron, for instance here, with an electric field or with the chemicals, then the signal travels all along the way. And then when it arrives here, neurons have rested for sufficient time to fire again. So basically you have a continuous activity on the system. And then playing with the total length of the pattern, you can design a clock. Okay? Something very funny that works very well. But my colleagues, advisement, are working on that. They also build and OR gates, and now they're trying to make a computer based on neural functions. Okay? But we want to do something different, and in our case, we are trying to put neurons on the valleys or on the tops of this topographical mode. In this case, we managed to put them on the top, you see. But the funny thing is that if you compare with the two-dimensional culture of the beginning, you realize that neurons really have a very complicated circuitry. Okay? And of course, this is one kind of configuration that is more or less random with a complex pattern. But here, our idea is to put neurons only on the valleys. So here we have clustered neurons that will be very strongly connected, and then we connect through these lines to the neighbors. So basically, we are trying to make a small world connectivity intentionally. Okay? And this is very funny. So I want to show you an example. So this is the behavior of this typical two-dimensional culture. That basically all neurons fire at unison. Okay? And if you explore what happens in these pattern cultures, then you see that there is a much slower wave propagating, and then it goes very fast, and then it's slow again. So the dynamics is very, very rich compared to the typical two-dimensional culture. And then let me show you an example. You will need a bit of time to. Okay. You see? It's a wave passing right to left. Now again. And there is another one. Do you see? That is not completely on-off, but there is a wave traveling along the system. Well, anyway, do you see it or no? Yeah? It's difficult, no? You see?
You are more used to it.
Could you follow the wave?
Yes, I can follow the wave.
Could you mark for us?
Si lo muestras por el puntero.
Ah, okay. Ah, sorry, yes. Yeah, it's going this way, and now... Okay. You want me to pass it again? So now it goes like this. You see? And now this way, I think. Oh no, sorry, again the same. Yeah, you need some time, I think, to get to that. But basically, these are our ideas so far. So that was one example.
Those were numerical simulations, or was it?
Those are experiments.
Experiments already.
This is an experiment, and this is another experiment. It is the one with this star. But here it's more difficult. I mean, you will see that the neurons at some moment fire, but it's very difficult to see a wave. But we are working on that. And you see? Did you see firing? Yeah. Okay. It's difficult to see, I know. But it's because we tried very hard to find a way to represent it very nicely, but it's very difficult. Okay, we have to improve this. So basically, what is interesting for us is that we can play with the connectivity of the network, and then we change completely its dynamics. So basically, what now we want to do in Barcelona together with you and other people is try to find out what's going on here. What is important of the connectivity to induce some special dynamics in the system. Okay? But I think it's something very, very beautiful. And somehow it goes in the direction of the title of the talk, and is, can we really build a brain at some moment? I mean, can we play with such refinement in the location of the neurons, like the genes do, and be able to build a culture that has properties of a brain? Something that is very interesting, and we are working on that somehow. And to start with that, I can show you, just to finish, a couple of numerical simulations. So at the moment, what we're doing is to try to understand this correlation between the connectivity of the network and its activity, is to try to prepare simulations where we accurately describe the behavior of the neurons and the axons. So in this case, for instance, this neuron launches an axon, the axon touches the wall, and then the axon follows the wall, and then goes straight. Okay? So we really try to simulate the behavior of the axon when they move in a substrate and find objects. Okay? We also simulate the genetic tree, which is basically will be a cloud of probability of connection around the neuron. So basically, if a neuron has an axon nearby, it will make a connection. Okay. In this case, we ignore completely the genetic tree for clarity in this picture, but basically these thin lines that you see here are actual connections between two neurons. Okay. But the important thing is that we can really make very detailed simulations, and this is, in this kind of patterns, the connectivity that we have. And to finish, I will show you a couple of simulations. And as you see, the nice thing about this kind of patterns is that we can run the experiment with the pattern and then use that pattern for the simulation. So we can compare exactly the two systems, and then we can tune the simulations until we can reproduce, well, as close as possible, the experimental results. And these are finally the last movies. So we excite the network up there, and you see the activity propagates now basically along the ring, and also using these nice connections in the middle. And in this case, that fired spontaneously before the signal from the others arrived.
So what's red?
What's that?
What is the red color? I mean, there are some neurons that have red color and then become black.
Okay.
So what is red? What is the?
Yeah. Okay. So the red are the neurons on the bottom,
yeah, but
and the white is neurons that are firing.
Right, but sometimes the amount of red decreases in some places. Seems that they go dark. Is that possible, or is it just an visual effect?
No. Neuron just fire, and then after a while.
So a neuron could only be red and white.
Yeah. At the beginning, it was something blue, Yes, yes, and that blue is because it's the point where we excite the network.
Okay. Yeah. Why did the counterclockwise wave die out?
I don't know. It's okay. You expect it to be in a different way, yeah?
Symmetric.
Symmetric, yeah.
It
depends.
I mean, this is one event.
This is one event.
Yeah,
No, but
there's another one.
But I mean, we don't have, neurons don't have exactly the same number of connections. We have a distribution of connections, so each neuron has basically a different axon way of looking for neighbors. We also have a different connectivity.
And you have noise also.
We have noise, exactly. The identity tree is also different from neuron to neuron. I mean, we use biological data to reproduce the neurons, but neurons are not identical between them. And finally, this is a simulation with a square. You see it well, no? Yeah, more or less, yeah. So again, we excite there, and you see many neurons fire at the same time. And well, yes. I need to start to propagate. I will jump a little bit. I was supposed to very slow. I will do one thing. I will change the program. This is better. So now I'll put it faster. So you see, the front is there basically, but you see the neurons are lighting up randomly in other locations of the culture. Because we have also some noise in the system.
So those ones are not that they are connected because of a one action, they are just
Exactly, they're just
they are not
firing.
connected.
Exactly.
But in your photos of your experiment, you had the axons which went through the void places. Yeah.
Well, in our experiments with similar pattern, well, yeah, we are now using GFP to try to estimate how far the axons go. But according to our estimations, one axon here will go maximum there, but no farther. This is about 6 millimeters in total, the width of the system. Well, I don't know if experimentally we will have such a nice wave propagating in the system. I'm not sure. But we are working on that. But I think it's very, very nice. And that's it. And to finish, I just wanted to thank the people that is working on these problems. Apart from the people advisement that is later. Yeah, basically, Nuria and Mary are working in 2D cultures with this kind of patterns. Sarah is working in 1D patterns to try to characterize how, if we can really follow the front in a very simple geometry. Patricia is our biologist, and she is preparing GFP and image-staining techniques to get information, real information about the structure of the axons and the dendrites. And there we have a group of permanent collaborators that most of you know already. Jaume Casalamunt, that is our leading theoretician in the group, let's say. And Jaume is working together with Enrique Alvarez, that was also with me at the Advancement Institute. And then Javier is working on these detailed numerical simulations. And at the moment, that's it. Thank you.
Any comments or questions?
Is there any biologist?
I don't know. Are you a biologist? No.
Yes.
Yeah. You have two biologists here.
Well, maybe you know that it's very difficult, right, to get information about the connectivity between neurons.
Yeah, of course, my opinion is that when you want to translate the models or the experiments to the real world is complicated. Of course, it's the first step, and anything with small animals, for example, ants or other pieces of mouse, could be possible to step by step.
In one of the first graph, you had the distributions. It doesn't matter. You had the distributions of frequency or the pulsating frequency of the isolated neurons in the culture.
The excitability of the neurons. That was
excitability of the neurons. This morning we discussed, would it be possible that if you take, I don't know, 1,000 or 100 neurons with a frequency that are distributed according to that function, which was a Gaussian, and you estimate the time it will take to coincide in their pulsing in a given window, certainly, would that probably match with the experiment, which would mean that just because every sometime or every time period they have to overlap the firing pulses, and then you will get some real light that you see. And it's probably not much due to the connectivity, or you think the connectivity is very, very, very important for us?
I think the connectivity is much more important, because that is the excitability to an external perturbation. I mean, not the natural excitability of the neuron.
Yeah, I know, but you have the excitation there.
Yeah, yes. But I mean that when you measure spontaneous activity, the system cannot naturally generate.
I agree with you.
That.
I agree. Yeah, Emilio?
I see here at least two levels of connectivity. Here you have you say that the axons can only go 1 millimeter away from the neuron, and then those big patches you have shown with patterns on them, it was larger. So you have the connectivity among individual neurons that you really don't show on the plots. And then is this labyrinth path you impose on it, or the small wall connectivity. Yeah. And then the links between places are not single actions, but bunches of actions.
Exactly. Yeah.
Is that what happens in the brain? I don't know if there are. In our brains, I think there are actions that go all the way to the So what is the important scale on the individual connection between neurons, or the connection between bunches of neurons?
Okay. That's a good question, because actually the brain has everything. Has a combination of all different kind of architectures, and depending on the circuitry is closer to one or to another. So in one place it's more homogeneous, in other is more wall or power law. So there is a lot, but on the other hand, apart of this, there is a hierarchical structure in many parts of the brain. So you have a group of neurons that are connected with other group of neurons, and all that group is already connected with other group of neurons. So you have all kinds of configurations that you can expect in network theory. It's extremely complicated. So for instance, each part of the cortex that is doing something specific, you can see as a huge aggregate of neurons. But inside that, there is also some structure. Okay. For instance, the motor cortex is very different from the visual cortex, or from the auditory system. It's very, very different. They have many similar architecture, but they're completely different in the behavior and in the way neurons are connected.
Do you have any way to control this hierarchy of connections? I see that they connect spontaneously between neighbors, and then the blobs connect between them. But I don't know if you can really force long-range connections with some control.
We have ideas about that. So one of our ideas is to use microfluidics. So we will combine this kind of pattern networks that you have there. Yeah, we'll put below a microfluidics platform. So what we will do is to put a gradient of chemicals that axons like. So we can force or have the axons to grow to much farther distances. Okay, we already tested that. Those compounds are called neurotrophic agents, and we can use neurotrophic agents, for instance, to force one area to have much more connections than normal, for instance, or use a flow of this agent to help an axon to be very, very long, for instance. Actually, in our body, we have very long axons. I mean, typically, from a sensory neuron in my finger up to the brain, it is a single axon. Okay, that is about 1 meter long. So actually, you can do it. The problem is that neural cultures, of course, don't have all the battery of genes that are helping them to do exactly what they have to do and to guide them correctly to the rotations. And you can trick the neurons in cultures to do something similar.
For the results you showed, how critical is the geometric size of these structures that you choose? And did you vary this size of your geometric structures? Because they will also let's say, determine the coupling strength, right?
Oh yes, exactly. Yeah. We're playing. I mean, at the beginning, we used just a random distribution of squares, but now we start to do some serious statistics. So we are using, for instance, different distributions of the size of the islands or the size of the patches, and also the distance between them, this kind of thing, also the connectivity in diagonal. And so now we are trying to really explore the possibilities that you have to prepare a distribution of known statistical properties. For instance, that these patches here are distributed power law or exponential, for instance, and then see how that influences the activity of the network. We are working on that.
Any other question or comment?
Well, but sorry, typically, due to, let's say, technical reasons, the minimum square that we can make is about 10, 20 microns on size. This means that actually you can put a single column with a neuron on top of it. You can go to that extreme, or even you can make very small channels, like this one. You can make very small, and only an axon can go along it, and no neurons, for instance.
Any other question or comment? If not, thank you very much again.